This is the latest in the series of workshops that started off as a one-day event a long while ago now. These workshops proved very, very popular, and so we've decided that we would carry on and have one per month. Just a few notes. This we'll try and have a break after about 1 hour, just to give people a chance to have a walk around. If you have any questions or anything like that, please put them in the Q&A rather than the chat. We will be monitoring the chat and pass on any questions to the people who are presenting. If you're in full screen mode presenting, unfortunately, you can't see the Q&A. So this latest workshop is courtesy of the Oxford Protein Informatics Group. And I'd like to hand over to Charlotte Dean to introduce the presentations. Over to you, Charlotte.
So good afternoon, everyone, and introduce myself to start with. So my name's Charlotte Dean, and I head up the Oxford Protein Informatics Group. And with me today, who will actually be doing all the heavy lifting, are Fergus and Matt, who are both members of my group and have been for a very long time now. So they probably know more about anything I'm going to talk about than I do. And I'm also now going to give a very quick apology. Unfortunately, I'm going to disappear as soon as I finish my talk, I'm afraid, because I have to go into a different meeting. But as I said, I think the other two are far more capable than me about describing what we do. So we're going to talk about one specific aspect of the work in the Oxford Protein Informatics Group, which is around our antibody modeling tools. I suspect that not everybody here works with antibodies on a day-to-day basis, particularly given that we're not in an antibody workshop here. And I thought I would start with just a very quick introduction to antibodies. And the other thing I suspect is that you can't have avoided hearing about them over the last 18 months. Antibodies are a vital part of your immune system. They are what the vaccines for COVID are trying to stimulate, one of the mechanisms they're trying to stimulate, to try and give us an immune response against it. They're what fights off invaders. It's been estimated the typical human is capable of producing more than 10 to the 12 different antibodies. And these kinds of numbers mean we can't just sort of hit it really hard and try and compute everything, because our computers aren't big enough or fast enough. And they recognize these potentially harmful molecules, which from now on I'm going to start talking about antigens. So that's the binding partner of the antibody. And they either inhibit the antigen themselves or recruit a different part of the immune system to deal with it. Now, the other part about antibodies is not only are they our natural defense, they've also become very prominent in terms of other things. So I've already mentioned the concepts around vaccines, but also they can help you diagnose exposure, whether you have been exposed to COVID. But perhaps most importantly and most interestingly developing at the moment is the development of effective biotherapeutics. Because antibodies can target specifically and with high affinity, as far as we can tell, almost any antigen, then they're fabulous as a drug. There are about 80 approved antibody drugs at the moment. And the thing that's very important to remember about them is they're entering clinical study at an incredibly rapid rate, because actually they're much quicker to, if you like, conceive of and build than a traditional small molecule drug. So it's much faster to get to an antibody that binds to your target of interest and binds to it specifically than a small molecule. Now, they do have downsides. They're obviously quite a lot larger. So if you're going to administer them, you're generally talking about things like injections rather than a small white pill. So that means they're probably not suitable for every kind of medication, but they do have advantages in terms of the speed that they can be developed and the flexibility with which they can be developed. So this is a quick primer around antibody sequence and structure. On the left-hand side of this slide in the kind of block diagram is the structure of a full human antibody. It's made up of four chains. So it has 2 copies of a heavy chain and 2 copies of a light chain. The part that we're going to talk about exclusively today is called the FV, and this is the fragment variable. It's the bit at the top end of this where the binding to the antigen happens, and it's made up of a VL chain and a VH chain, so part of the heavy chain and part of the light chain. And on those are a set of things called the complementary determining regions. These are loops that are primarily responsible for binding to the antigen. Now, these loops have a lot of sequence variability, which is why they are able to do these different types of binding. So the two things to really emphasize on this slide is that genetically antibodies are made up from lots of different pieces of genes. So you can see the VH there is made from a V gene, a D gene, and a J gene. And it is that combining of those pieces, and then if you like, the direct and on purpose encoding of errors when you do that, meaning you get huge diversity. And then on top of that, you have processes called somatic hypermutation to try and increase that diversity and binding affinity. And then the second part of this is those regions involved in binding are on the tips of these two domains. So it's actually a relatively small site we're thinking about here, but it is also between two chains. So the orientation between these two chains and how they sit with respect to one another is another important feature that we have to be aware of when we're doing this. The big step forwards in antibody work over the last few years has been in the area of next generation sequencing, so B-cell repertoire sequencing. What that has enabled is the ability to take samples from an animal, in this case a human is my very bad picture there, and see what antibodies are circulating in their body. Now, the issue here is that the circulating diversity is estimated to be about 10 to the 9. These snapshots always sound enormous when people report them, but even the biggest ones are really only in the hundreds of millions. And that's, as you could tell, is only a tiny percentage of what's actually circulating. So it's still something you have to estimate and be aware of, that you haven't got everything. And the second issue with this data, which is important to be aware of as we step through using it and thinking about it, is currently, though this is changing at the moment, most of the data is only of the VH or of the VL, not of that paired sequence I was talking about. So you don't know which VH goes with which VL. But the exciting things you can do with this data are things that relate back to what I was talking about before. You can look at somebody pre and post immunization and say what's changed in their repertoire. I can say what's different about a human repertoire from a mouse repertoire. And that's incredibly important. If I want to make a safe therapeutic, I need to know that I'm not giving someone something that looks like a mouse antibody. It needs to look like a human antibody. Otherwise, my own system will knock it out. So you can see all of these things. You can also use these repertoires to go fishing for things which might be exciting or interesting antibody targets or drugs. So now on to the work that we're going to share with you. I'm going to start with our databases. And I think it's very important to kind of start here with the data you have, because it's really this data that allows you to do so much. So this is 3 of our main databases on here. The first of these is observed antibody space. This is a collection of those sequence data sets I was just talking about. And the important thing here is that we have to clean that data. I don't know how many of you have had to deal with data, sort of genetic data or data from next generation sequencing. It's not a particularly clean data type as it is produced. So here we sort it, clean it, annotate it, and make it easy to use in the antibody space. And that data underpins a lot of what we do. The second database that I would say underpins almost everything we do is the structural antibody database. And this is a fully automated updating collection of all publicly available antibody structure data. And the reason for doing this, and I'm sure many of you have used the PDB, is if you go to the PDB and you type in the word antibody, you don't get all the antibody structures. So we needed a different mechanism to allow us to do that. And once again, we clean it and curate it to make sure it's consistently presented. And this is all done under an automatic system. There's just over 5,000 structures in there. And to give you an idea on this, the last time I gave a talk about this, which was back beginning of June, about June the 9th, so I updated the slide this morning and looked at the number from June the 9th, it was about 150 structures lower than that. So this number can move quite fast, and at the moment it's moving very fast because there's a lot of interest in antibody structures at the moment. And then a third database that is very useful to us and that we've built, it's self-updating, it does need work from us, it's not fully automatic yet, is a collection of all of the immunotherapeutic variable domains that are ready, if you like, to be used as therapeutics. So through a certain stage, we harvest these from the World Health Organization. There's about 650 entries in that database. And that helps us have a description of what makes a good immunotherapeutic. And Matt and Fergus will come back to talking about why it's so important to understand that. If you think in terms, it's very useful to understand what makes a good small molecule, you know, what properties make something a good small molecule is a general thing about things that might be possible to be good drugs. And in the same way, you can think about antibodies. Is there a description of what makes an antibody a good biotherapeutic as opposed to something you wouldn't wish to use? And another database I'm going to mention briefly, partly because we're going to use this as sort of our example for helping you to see your way through the tools, is CovAbDab. So this database, as of July the 9th, had 3,250, something like that, entries. And in here are things that bind to coronaviruses. Now, the vast majority of that data now, I think it's nearly 3,000 of that, are things that bind to SARS-CoV-2. And these are all antibodies that are experimentally verified to do that type of binding. And it makes it a very exciting and interesting data set to work in, not just because it has huge implications for health right now, but also for our understanding of how the system works and how you can predict the properties of things that will bind to a given target. Now, I've described sort of sequences and structures, but one of the big pushes in our group has always been to see the world through the structural lens. And the reason I want to do this is because I always think, well, if you're going to understand how something binds, it actually does it with its three-dimensional shape. It isn't a list of amino acids at that point. It isn't a sequence. It isn't a pretty picture. It's a definite three-dimensional shape. Now, that shape may be in motion, but it is a shape of some description. And the issue we have always had is something that this picture demonstrates quite well, which is, it's slightly out of date now because these numbers only go to March 11th this year. But we started this work sort of back in the spring of last year as CovAbDab started collecting information, and we decided it was really important to have that information freely available to the entire community in a good format. And what you can see is the number of antibody structures, if we look at the picture on the antibody structures in blue. Now, people are desperately solving these, probably faster than any other category of antibodies we've ever had, and they're working incredibly hard at this. But at that point, we were talking about 100 structures solved. Whereas if we look at the number of sequences, full sequences of antibodies that we knew bound, already on that date, we were at sort of 2,300. And so we're never going to keep up with our sequence information in terms of solving structures computation experimentally. So the big thing for us was we can close that gap computationally. And one of the ways I'd like to emphasize this is actually modeling antibodies is, I don't want to say easy, but we're pretty good at it now. But also excitingly, lots of the other developments that are improving structure prediction, I'm going to guess most of you have heard at least something about AlphaFold over the since December last year, also influences us here. It isn't a direct, I'd use AlphaFold to predict antibodies because it has particular properties that might make it not suitable. But the technologies that are being developed there are impacting our ability to predict here and will mean that computationally closing this gap is totally realistic. I can have good models for this. So what does it mean if I can move to this kind of structure-based world and work in that way and think in that way? So I'm now going to give you a quick wraparound of our tools that all come out from that. And then I'm going to hand across to these guys to let you sort of play with them and see how they work. So I'm going to start top right on this slide with something called SARB+. What SARB+ is for is just very rapidly annotating those massive repertoires of sequences with structural information. Don't try and build full three-dimensional models, it's too slow. Because if I tell you there's over 1 billion sequences to build models of, even if your modeling program builds a model every second, and most programs aren't that quick, we think we're pretty quick and we're down in the sort of 20-second realm. You need a very, very big computer to do that, and it's probably not worth the effort. So instead, we have strategies to, if you like, build enough of the model or understand enough of the structure to take information out. And I'm going to go across the top of the slide. So I'm going to go to ABANGLE next. There, that's looking at and predicting, but also more importantly, characterizing the angle between the two parts of your antibodies, the two chains. And this is important in terms of how those might change as well upon binding. So understanding that conformation and being able to compare it to other similar structures. And then a whole set of tools, so A-Body Builder, Scallop, Pairs, and Sphinx, and they are all about being able to build these three-dimensional models of antibodies. And we are continuously working on improving those. There's some tools that we're just about to publish, so unfortunately we can't show you today around improving our predictions there, but they'll all be part of this suite, so they'll appear on our website in the very near future. And down to the bottom left, DLab is I think a very explaining name, but basically this is moving to the idea of using deep learning to predict whether a given antibody will bind to a given antigen. So just give me an antigen you're interested in, tell me where you want the binding site to be, and I will just dock antibodies against this, and I will say whether it will bind or not. And using deep learning techniques very similar to the things that have been used in the small molecule world. And then we have HUMAB. So this is for the humanization of antibodies. So anything that I give a human, if I'm giving them a protein of some type, I need to make sure I won't raise an immune reaction in the human. So if I give you something that raises an immune reaction in you, so it is a non-human protein, if I give you a mouse antibody, it won't be a very effective drug because your immune system will take it out. And even worse than that, your immune system may overreact to it and cause you further complications beyond any disease I was trying to cure. So humanizing antibodies and trying to make sure they are human before we put them in any humans is a really important task along the way. Anarchy. This is one of our programs that won an award for one of the worst names you can give a program. I happen to like this name quite a lot. It's a numbering program. So Fergus and Matt will talk a little bit about having numbers on antibodies is a really useful way of being able to describe them. So it's really a tool for kind of describing your antibody and allowing you to do consistent analysis. In the bottom right, paratyping and obligity. These are two tools for trying to predict if two antibodies which are sequenced very dissimilar will bind to the same binding site. And this is a really interesting problem because it's relatively, in fact, very straightforward. If two antibodies are very sequence similar, just like you see in general proteins, that means they're going to look very alike and they're going to do very similar things. But if that antibody, for example, has some very bad, it's very difficult to humanize, you can't use it. So what you want is something that will do the same thing, but its sequence is potentially very, very different. And these tools are built and designed to do that step. And then finally, TAP. So this tool is entirely designed, it's called the Therapeutic Antibody Profiler. But what it's designed to do is not work out if an antibody binds, but work out if it's what's called developable. The easiest example of developability is if I want to have an antibody to use as a biotherapeutic, I'm going to keep it in a bottle in a fridge at some concentration. I need it not to aggregate. Yep. Now, that's a property that's very important, has nothing to do with binding, but if it is something that aggregates very easily, it will be an awful therapeutic, regardless of its binding properties, because I just can't deliver it to patients in a safe way. So that was a very rapid walk around of some of the tools you can find on our website. Matt and Fergus will take you through lots more of these. I'm just going to very quickly mention that we have suites that revolve around nanobodies and also TCRs. So if you have deep interest further into biotherapeutics and other types of these, feel free to ask Matt and Fergus more about those as well. And I have to finish by pointing out that I don't actually do all of this work. I do a lot of the waving and smiling and talking about it. I promise I think about it quite a lot as well. But the guys in this photograph do most of this work, and particularly people in the group like and who have left the group like Claire and Catherine and Alex, and people like Matt and Fergus and Eve and Sarah have been deeply involved in all of this. So I think I will stop there and hand over to my colleagues who will take you through everything else.
Then, so just before I get started, I'm going to do a very brief run through of what the structure of the rest of this workshop is going to be. So we've just had a roughly 50-minute talk from Charlotte. It gave you sort of a very high-level overview of the databases and the software suite that we've developed in the group and that we're going to be talking through today. So for the remainder of the workshop, what we're going to do is, there's going to be a 10-minute break halfway through the workshop. So between now and that break, Matt and I are going to sort of walk you through a case study of starting from one of our databases, identifying some potential candidate antibodies for SARS-CoV-2. And so we're going to take you through how we might mine that data, and from that data really whittle that down to just one or two sequences of interest. And we'll talk about how we can tie together the sequence-based data with the structural databases and how we can use that to really get a good grounding for trying to develop an antibody therapeutic. And then we'll sort of take you through doing some structural modeling of the antibody sequence that we arrive at, and then doing some functional profiling of the model sequence to really try and construct a structural hypothesis for how this antibody that we've selected might bind, and if there's anything that we might be concerned about or need to engineer further or optimize. And so we've provided you in the data pack that you were provided for this workshop. We've provided you with sequence and structure files for all of the data we're going to use today. There's going to be a PyMol session at the end of this first hour where I'll walk you through the structural side of things. We've provided a PyMol session file if you want to open that up pre-prepared. In the cheat sheet that you're provided with, you'll find a description and walkthrough of everything that we're going to do, as well as instructions for replicating the session yourself if you'd like to sort of work through that offline as well. So please feel free to follow along if you want to work on split screen, but at the same time, if you want to just focus on us walking you through for the first hour, then that's absolutely fine as well. And then there's going to be a 10-minute break, and then following that break, what we're going to do is we're going to have sort of a more open-ended exploratory play with a couple of the tools on our web server, which are really geared towards, Charlotte alluded to this idea of developability. We're going to introduce you to two of our tools that explore that concept and have you take an antibody sequence and play with those tools and get a feel for what can we say about the developability of an antibody starting from its sequence. And then we'll have some time at the end to wrap up and answer any questions. Okay. So this is really bringing us right back to the very start of what Charlotte was saying. If we think about the data that are available to us, really the largest quantity of data that we have available comes from next generation sequencing, where we have this very large selection of antibody sequences that have been taken from a great many individuals, and next generation sequencing has really led to sort of an explosion in the amount of antibody sequence data that's been available for and NGS really allows us to query the enormous natural diversity of immune repertoires, but the resulting data sets that you'll find across the literature are quite scattered. They're often very heterogeneous, and they often present things in terms of nucleotide sequences rather than amino acid sequences, which is what you really want to work with if you really want to start sort of building structural hypotheses of what these proteins look like. So to solve this problem and get this data into a format that's useful for people thinking about proteins, we developed the Observed Antibody Space, or OAS Database, which brings together a large number of NGS data sets. And Charlotte mentioned this idea of numbering amino acid sequences. This is where you apply a standard numbering scheme that identifies particular structure parts of the sequence. For example, certain ranges correspond to the framework regions, which are sort of fairly constant across antibodies. Other numbers might correspond to the CDR loops, which is where this great variability in affinity and specificity arises. And it gives you a very standard way of annotating and comparing sequences. So we've added these annotations to all of these NGS data sets that we've acquired. And we've structured this in such a way that all of the data are available as sort of distinct data units with metadata sort of characterizing where that data came from, what you can expect to find in it. And this allows you to do things such as, for example, tracking samples taken from an individual at different points across a vaccination study to see, how did their immune repertoire respond to the vaccination. You can compare before and after vaccination. And so I should also mention, in the cheat sheet that you've been provided with, towards the bottom, there are links to all of the databases and tools. So if you want to click along to any of those, you can find the links in there. So what we're going to do is talk through how we can extract some SARS-CoV-2 antibodies from this data. So if you're following along, if you navigate to the OAS database, this is the front page that we'll be met with, and immediately you'll see that, this is something that Charlotte mentioned. We do curate both unpaired sequences, so just the heavy chain or just the light chain, and also paired sequences, so the data has actually identified which heavy chains go with which light chains. Now, I should say that the overwhelming majority of NGS data that's currently out there is unpaired sequences. So the quantity of paired sequences that we actually have access to is almost vanishingly small compared to that of unpaired sequences, but the data are there. But for the purposes of today, we're going to focus entirely on the unpaired sequences. So if we now just click on the unpaired sequences, we can bring up the OAS search function. And immediately what you'll see is there are a great many fields that you can specify in order to narrow down your search through the database. So just to give you a few examples of some of the properties that you're able to query the database by, you might want to query only for heavy chains or only for light chains. So we can specify the chain type. You might be interested in the age of the individual. For example, say you want to explore differences in immune repertoires between young people and old people. You might want to explore immune repertoires associated with an immune response to a specific disease. So for example, today we're going to be looking at SARS-CoV-2 data, but lots of data sets have been annotated with lots of different diseases. So there's a great diversity in there. You might be interested in different B cell types. You might be interested in exploring, as I mentioned, immune response to a vaccination campaign. So you can pull out only studies that have been annotated as being part of a vaccination campaign. One of the obvious things you might be interested in is I only want human data, or I want to compare human and mouse data. So you can search for data according to the species. And finally, you can also specify whether or not you only want to obtain data where that data has actually been mapped onto a specific subject. So in some NGS studies, you won't actually have information about which individual in that study. So say you have 3 people, some data sets might not have the information about which individual that sequence came from. One of the things that we've done is annotate the data in such a way that you can filter it down to only data where you have an individual. And again, this can be really useful for comparing, for example, how did this individual's immune repertoire evolve over the course of a vaccination campaign. But what I'm going to illustrate now is if we just search through only for data associated with SARS-CoV-2, what we'll map is a number of what are known as data units. And each of these data units is a collection of sequences that are uniquely identified by all of the metadata parameters defined individually. So in the results for SARS-CoV-2, what we can see here is for each of the data units, we record what study did it come from? So what publication is this in? We record the number of sequences present in the data. And I should point out, if you're not familiar with NGS sequencing, that a lot of this data is very redundant. So you may have a data unit where you have many copies of the same sequence. And this is in fact a good thing. If you observe the same sequence many times, then that gives you great confidence that the presence of that sequence in the data set is not just the result of a sequencing error. If you come across a sequence that occurs precisely once in precisely one data set, then you have to start thinking about whether that is actually a sequence found in the repertoire or whether it's the result of a sequencing error. Obviously, in this case, we're dealing with human data. The vast majority of the data in this case is also antibody heavy chains. We can see that all of these data units have been annotated with a specific individual, so we can already start thinking about, do we want to compare individuals? And finally, we also, again, we annotate sort of the time at which it was taken. So again, you can track how did this individual's immune repertoire evolve over time after exposure to coronavirus. And so you're able to, you can download any of these data units individually or bulk download the entire set of data. And so again, just to break this down in more detail, for a single data unit, you can click through to a summary of that data unit that will give you sort of all of this metadata information. And this really, by sorting the data in this manner and annotating the data in this manner, it really sets it up nicely to be used in these sorts of comparative studies. I want all human data and I want to compare it to all mouse data, or I want all data associated with this disease, associated with a vaccine campaign that has date ranges for specific individuals. It really allows you to make these sort of rational decisions about the data that you're obtaining. So that's sort of at the very high level, the bulk data that we have available to us. But now we want to think more about how can we take this data that may consist of hundreds of thousands, millions maybe of sequences associated with SARS-CoV, with immune repertoires for people infected with SARS-CoV-2, and really pare this down to some interesting non-redundant sequences that we might want to actually explore developing as therapeutics. And for this, we're going to introduce the idea of convergent clones. So really, again, the sort of structured nature of the data allows us to explore this rationally. So we've sort of done this beforehand for you, because this is a process that can sort of take a little bit of time to really go through and get right and really arrive at something sensible. We've provided you with the sequences that we arrived at the end. This is really to give you a feel for how you would approach this problem starting from this data. So the first thing that we want to do is, for each individual in the data set, so now we're sort of restricting ourselves to one individual at a time, we want to take their immune repertoire and identify what are known as clonotypes. These are groups of sequences with common ancestor. So these are sequences. Charlotte mentioned that we obtain the diversity of antibody sequences by combining VD and J, different VD and J genes. And it sort of allows the body to go from this tractable initial step to an enormous diversity of sequences. And so in identifying clonotypes, we're looking for things with a common ancestor. So we group the sequences by their V and J gene annotations, which are provided by OAS. And then given those groupings, we then, for each grouping, cluster them by the CDRH3 identity. I recall that the CDRs are the region of the sequence that is responsible for antibody binding. And by performing this grouping and clustering, this gives us a group of sort of distinct clones for that individual. And what we're really looking to get out at the end is, in practice, we have a set of sequences for which we have a distinct V gene, a distinct J gene, and a distinct CDRH3. And this really gives us the groundwork for sort of representative sequences from this one individual. And so from there, what we can then start to do is identify clones present in multiple individuals. So say in our example, we did this for 3 individuals. And what we want to see is, are these clones the different immune repertoires will naturally converge upon in the presence of the antigen, despite being from different individuals. So we have this idea of sort of convergent evolution, even if they potentially start from different initial conditions. And so this introduces the idea of what's known as public clones. This is a clone that will naturally be converged on in multiple individuals. And so we can take all of these clones from our different individuals and then just pick out ones that are present across the different individuals. And so then what we can do from there is say, okay, we have these sequences that we've selected from OAS associated with SARS-CoV-2, but we want to then know, all that tells us is that these sequences were present in the immune repertoire of multiple individuals with SARS-CoV-2. It doesn't directly tell us that these correspond to antibodies raised against SARS-CoV-2. They were just present in the repertoires. So what we want to have a look at is, for example, are these clones also present in data sets not associated with SARS-CoV-2? So if you do have a sequence that is also present in these other data sets, these are likely to be sequences that are raised sort of as part of a general, general disease response. And these are what are known as bystander clones. So they're raised as part of the immune response, but they're unlikely to be specific SARS-CoV-2 binders. And so again, mining through the OAS data and applying this to our clones, for this case, we identified 2 distinct convergent clones. One, clone A, was found only in SARS-CoV-2 datasets, whereas another, clone B, was also raised in response to several other diseases. So you've immediately pared down the data quite a lot. And so for the rest of this workshop, we're going to be analyzing and modeling clone A. So this is the one that is likely to be specifically a SARS-CoV-2 binder. And so Matt is going to talk you through, we've gone through the process of identifying from the sequence data our convergent clone. What can we then do to take this sequence and turn it into a structural hypothesis for how it might actually bind? So over to you, Matt.
Thanks, Fergus. Yeah, so we've seen here how our big database of human immunoglobulin sequences that have been pulled from lots of different people in disease states can help us hone in on particular sequences or lineages, as we call them, that are commonly expressed amongst people with a particular disease state. And here we found that this clone A has been seen in 3 different individuals, all of which were recovering from SARS-CoV-2, but we couldn't find this same sequence in, for example, healthy people whose repertoires were just kind of on standby mode looking for infection, or in people who were recovering from other diseases. So this has really given us a kind of statistical indication that this might be a sequence that can actually bind to SARS-CoV-2. And then from this, what we now want to do is look, given all the other information we know already about sequences that can bind to SARS-CoV-2 and structures of things that can bind to SARS-CoV-2, can we get a molecular rationale then beyond this statistical indication towards a molecular understanding of how this sequence could bind and could go about protecting each of these patients? So what we've done here is we've just recapitulated some particularly important properties of the sequence. So the CDRH3, which is this hypervariable loop in the heavy chain, equates to this portion here, but we've just grabbed it out. And the V gene, which encodes the majority of the sequence until around this CA block here. So that portion of the sequence is to some extent pre-encoded on a V gene, which is a gene that you have in your chromosomes, the 58 gene, and has a standard sort of amino acid sequence associated with it. But of course, this express VH sequence might not be the exact same sequence. It might have somatic hypermutations at particular CDR points to have taken it away from this germline. But this is the closest germline that we can map it to. So it gives you a sense of what genetic origins this sequence was likely to have. And the J gene as well, which is the last portion of this heavy chain sequence, is this particular J gene as well. For some of the future work, we will need to consider.
Matt, I think we're still seeing Fergus's screen.
That's correct. Yeah. So Fergus will be flicking
so we
slides
can't.
for me.
If you're using the pointer, we can't see it moving.
Okay, so I'm just talking through the slide really at this point, but
okay.
I'll give cues to Fergus to point at things as helpful. So if you could point to the VL sequence, Fergus. So this is not something that's come from our repertoire data. This is something that we've burrowed from an in-house library of light chains that we know are compatible with this heavy chain germline. And this is useful if you want to start to build hypotheses about contiguous FV binding sites. So we've got the heavy chain implication from the repertoire data, but for now we're just borrowing a compatible light chain sequence that we'll use in some informatics later on. And it's highly likely this VL sequence, will need to be optimized in some way to achieve optimal binding. And you have this sequence, the heavy and light chain sequence, in a FASTA file if you want to play around with this data later. Can we go to the next slide, please, folks? So the first thing that I'll be doing is comparing this convergent VH clone to the database of coronavirus binding antibodies that we have to see if it looks in any way sequence or structurally like things that we know can bind to coronaviruses. So as Charlotte has introduced this database, I won't dwell too long on it, but essentially it's an attempt to collate all the information that we currently know about antibodies that can bind to SARS-CoV-2, but also related to coronaviruses such as SARS-CoV and MERS, and even sort of endogenous seasonal coronaviruses too. And to be included in the database, it needs to have a minimum of the CDRH3 amino acid sequence, so that very diagnostic loop sequence, and the genetics that sequence can map back to. But ideally, and most of the entries do have full variable domain sequences, so you could go and actually make computational models of these things, and many of them, and an increasing number, have solved structures as well, because there's as Charlotte alluded to, there's a huge effort to solve these things in complex with SARS-CoV-2, so we can understand the diversity of binding sites that exist on this virus and how they vary, for example, by variant. So next slide, please. So we can search the database in many different ways. We can go hunting for, say, a particular heavy V gene, which you'll see in the attributes column there. So we could go searching particularly for things of the 158V gene if we wanted to, or and only antibodies that have been shown to be neutralizing, or we can query the database by sequence properties, and this is what I'll actually be doing in the live demo. So taking parts of this sequence that we've identified as convergent and querying the database by sequence to see if we get any hits back, any similarities back. So this allows you to use this data, and it's all hosted freely available online in a way to suit you. But hopefully it will be explanatory for why we might want to do this in the live demo. We'll be feeding our convergent clone property in, and we'll be seeing if we can get any functional inference from the hits that are close to it in sequence to the CDRH3 loop. Next slide, please, folks. And from there, we'll also be looking at some of the solved structures of antibodies in complex with SARS-CoV-2, which will link us nicely into our other database, which is the structural antibody database, or SABDAB, which has now recorded over 5,000 PDB entries with an antibody structure inside them. And again, there's a whole range of ways that I'm not going to show you to search this database. If you're interested, you can do a similar type of sequence search the way we did with COV-ABDAB to go hunting to see is there a solve structure of something nearby in sequence to this clone, or you can hone in on particular CDRs, or you can search on orientation parameters, like Charlotte described, the AB angle tool, which characterizes the orientation between a VH and VL domain. You can do similar things where you only strip out the sequences that have a particular orientation property. And you can focus on nanobodies if you want, rather than antibodies as a general category. So there's lots of different ways you can use this data. I'll show you just one way in the context of this pipeline, but it's a very malleable database. And if we click through to the next slide, you can see the sort of readout that you get for each entry. So there's a metadata table that I haven't shown here, which tells you a lot of parameters about the antibody structure itself, but it also allows you straight away to download this structure numbered in some of these intuitive numbering schemes that Charlotte was mentioning via our Anarchy tool. So you can get it in an IMGT or a Chothier numbered format, or you can just get it as the plain wild type. And a nice new feature of our database is that it talks to all of our other databases. So it automatically detects, if you're just searching on SABDAB itself, if this is a sequence match for something that's in our coronavirus antibody database. So you can do the sort of inverse of what I'm going to do, which is hunting through COV-ABDAB to link to SABDAB. You could do the inverse of this very easily as well. And then this has all been quite based on sequence searching at the moment. So the next step, the next slide, will be kind of turning this into to structural searching, which is not that commonly done in the field yet, mostly because of its difficulty in terms of its tractability to get higher accuracy models quickly enough. But really substantial developments have been made in this field over the last five or so years, which really has allowed us to do rapid and relatively high confidence models of FV sequences. So we can really start to build up a case for how in 3 dimensions this antibody would fold into a contiguous recognition surface. And Charlotte stole my thunder a little bit for the next slide, which is to say how we currently do these things might not be how we do these things in the near future, because of technologies such as the AlphaFold Evo Former and Rosetta Fold as well, coming out with next generation structure modeling techniques that appear to significantly outperform existing techniques, although these are tackling the general protein structure prediction problem. And some of the ways that they have gained this increased performance, for example, by multiple sequence alignments, don't necessarily translate into immediate better antibody structures. We fed a few antibody structures into these pipelines, and they don't, as a whole FV domain, they don't tend to perform very well across the whole surface. What remains to be seen, though, is if they can do very well across portions of the surface. For example, in predicting the structure of the CDRH3 loop, which is very variable and is actually the really difficult thing to do in FV structural modeling, is getting that structure right. So it might be that some of the things that I talk through on the next slide will change in time, but this is currently how we go about doing it. So if we just flick to that slide, we use our tool called a bodybuilder, which is our modeling pipeline essentially. And we can treat the antibody modeling pipeline in a bit of a cheap way, really. We can make it a very modular protocol. So the framework region, which doesn't really vary in structure very much across antibodies, we can calculate its structure by borrowing the coordinates from a very sequence identical structure that's already been solved from SABDAB. Then for the loops, which are actually the bits often doing the recognition, we have to think a little bit more carefully about this. But the good thing is that we can do much of this now because of our template database is growing via homology modeling. So we can use our tool called Freed, and Freed is a way of taking our set of templates and considering their dihedral, kind of like their string of dihedral angles, and thinking, if I was to use these coordinates to model this sequence of this CDR loop, would the two be compatible, or would they likely not be compatible given this dihedral profile and the dihedral biases that amino acids tend to exist in? And this calculates quite rapidly a score that has to meet a certain threshold for us to consider this template to be a compatible loop template. And beyond that, we look for how well this sequence grafts, this sort of structural template grafts in the context of the rest of the antibody structure. So we're trying to minimize clashes here. And we can do that for all of the canonical loops, which are basically the CDRs that aren't CDRH3. And we can often do it for CDRH3 as well. In the occasions where we don't have a template that meets that threshold, we have to try some hybrid or ab initio approaches to model it in as more of a physics problem, or borrowing aspects from homology modeling and physics to make a decent guess of what that H3 loop looks like. And finally, we have a tool called Pairs, which uses Gaussian mixture models to predict which is likely to be the lowest energy rotamer of the structure. But in essence, what this is doing is we have our FV sequence, our heavy chain and our light chain amino acid sequence. And by going through a series of protocols, we can cast this in a structural representation, which is borrowing information from all these different places. And by using the structural representation, we can see things that we wouldn't necessarily see just from the sequence. And it proves to be, as a general principle, quite a useful representation to learn from in this field. So I think best now if I just start the live demonstration. So if I share screen on our tools. So just to check, Fergus, can everyone see this shared screen, which should be a Google Chrome instance with our CobAbDub database? Yep.
Yes.
Great. So here's the database live sitting on our website. Charlotte said it contains about 3,250 reads, and we can search in these different ways, either via attribute, which is one of the options I showed you on the slides, or what we're going to do here today is search by sequence instead. Now, what I've done is I've borrowed this CDRH3 sequence from our Convergent clone that was seen in lots of COVID patients. So this is the loop amino acid sequence of this very hypervariable loop. And you can paste in here a full VH sequence, or you can paste a VH and a VL sequence, or just a CDR3 sequence. So this is what we're going to do here. So just looking for close hits in sequence identity space across this database. So it goes away and looks for that. So it recognizes that we didn't input a full variable domain sequence. Instead, we only look for the CDR3. And below here, we have a table of the 20 closest sequence identity matches. So if we start to scroll down here, what becomes abundantly obvious where matches are in white and mismatches are in red is that actually this clone is not an outlier in terms of lots of other studies that have been performed into SARS-CoV-2. You can see some of the names here give away where they might come from. ION are sequences that have been released by IONTAS, which is a pharmaceutical company. COVOX was some internal Oxford sequences identified by Gavin Screeton. So there are lots of data points here that show there are sequence identity or sequence similarities between the CDRH3 of this Convergent clone that we've implicated statistically in terms of SARS-CoV-2 binding. And the nice thing about this is we can inherit and kind of get a functional idea for the properties of what this sequence might have just from these close sequence matches. So we have all of this metadata associated with each entry. So just going along the top row, with this antibody C005, it actually has an identical CDRH3 sequence, which is very, very unusual to just come across by chance. But we can see it binds to SARS-CoV-2, but does not bind to SARS-CoV-1. So it appears to be targeting a binding site that is in some way non-conserved across beta coronaviruses. And this follows through to its neutralization profile as well. We're also able to locate the binding site to somewhere in the receptor binding domain because the study which released this data actually expressed this antibody and did some competition assays and found that though this sticks by ELISA in the receptor binding domain, and potentially if you go hunting in this the associated literature, you might get a more precise binding region as well. We can see it's come from human patients, which is consistent with how we've obtained this Convergent clone of interest. It has the same genetic profile as our antibody. It doesn't have a solved structure, unfortunately, but we have pre-built homology models for things that don't have these structures. We have literature sources to go hunting back for this information. So we can inherit, or we can infer a lot functionally just from, oh, sorry, I think I just went back there by mistake. Let me just skip forward to that. We can learn a lot functionally based on the matches to this COVABDAB database. Talking about structures, if we scroll down, actually, we notice that while for many of these, they don't have a solved structure, we do actually have some solved structures in our database. So this entry here, S2E12, which has a 75% sequence identity, 75% sequence identity match to our convergent clone of interest, actually does have some solved structures here in the PDB. So the nice thing about solved structures is it might not just tell us about the structure of the antibody itself, but it might tell us how the antibody talks to the virus. So we know it probably talks to the virus or binds in the receptor bind domain, but we might not know molecular details or how it really fits into the receptor bind domain. So this information would be really useful for guiding docking studies or for trying to understand which bits of the sequence must be kept convergent. So what I'm going to do here is pick this first PDB entry and go through to our structural antibody database and see what this structure looks like and what it could tell us. So we can see it's solved by electron microscopy, which isn't ideal. Usually we tend to believe X-ray crystallography structures a little bit more in terms of capturing all of the electrostatic or non-covalent interactions that are going on in the binding site. But on the other perspective, it is actually very useful because it can tell us things about bigger structures. So we can understand how maybe larger fragments of antigen can interact with this antibody. So maybe the full spike domain rather than just the truncated RBD. And you can see this in the structure visualization down here. This is actually a massive structure. So we have the full spike domain, in fact, the full spike trimer here, bound to 3 equivalents of this antibody. So this structure is actually quite interesting for us because we can hone in and see really how is this antibody likely to be doing its binding, this convergent clone likely to be doing its binding in the context of the antigen binding site. So we can see it's true in complex, and it's about 3 angstroms, which is towards the upper limit of what we like, but it's good enough for this demonstration. Other bits of information we can find out about this are the basic genetic properties of each of the Fabs that are represented in the structure. And as I said before, links back to the database. So you can see here, this is linked back to an antibody that's present in COVABDAB, which is the inverse of how we got here. And ordinarily, although you have this in your structure pack, this would be where you would download the IMGT numbered structure, our preferred numbering scheme, to start doing some structural informatics between it and the convergent clone. So that is going to be our true positive data. So this is a structure of something we know binds to SARS-CoV-2 in a particular binding mode. But what we want to compare, if we want to do the structural comparison, we need to get a sense of what our convergent clone looks like structurally. Now, clearly, we don't have a solved structure for that. So we're going to try and build a solved structure by using our bodybuilder homology modeling pipeline. So in here, we have the space for the heavy chain and light chain sequences. So I'm just going to trim out the heavy chain and the light chain we've borrowed, remember, sequence that are associated with the convergent clone. You can recognize it's CDRH3 in here, sitting there. You can give it a job name. You can specify PDBs you don't actually want it to use if you don't trust any particular ones. And we're just going to keep these set to the default. So we're going to be comparing across the IMGT numbering and using modeler. And so we can start this modeling process going. Unfortunately, as you might expect with a lot of other structures around this space, it's going to be able to homology model each of the loops, which means we're going to get a structure prediction relatively quickly out of a bodybuilder this time. So no waiting around for the ab initio steps. So just going through this. Okay, so it's already finished. There we are. So we can see the different templates it's used for the different loops, which itself could give some functional inference if these structures, for example, have been implicated in SARS-CoV-2 binding themselves. And then you can view the model just to make sure it makes structural sense. There's no obvious clashes. All the loops here look like they are they're not bumping into one another. We can pick out different representations for different bits of the structure, like secondary structure, so which bits of the alpha helices or the loops. But we'll just stick with the domains. And this quite long loop is our CDRH3. It had this cysteine-cysteine motif within the sequence, you might have noticed. And the fact that these two cysteines are in close proximity to one another, again, bolsters confidence that whichever template it's used for the modeling of this more variable loop is quite sensible, because we've recapitulated a disulfide bridge, which we would expect to be a pinch point of the loop structure. So everything really checks out here. So if we go back to our main results page, we're able to download this as a PDB format, which has the IMGT numbering because we selected that from that before. So this has really demonstrated how we can go from a convergent clone of interest, hunting for sequence similarities and maybe structural similarities to things that have been solved to bind to SARS-CoV-2. And now Fergus will talk you through aligning this model and the structure in a molecular viewer to see, in the context of the antigen binding site, does this look like a realistic or a sensible structure to be a SARS-CoV-2 binder. So I'll just stop sharing my screen here, and Fergus, you can take over on the PyMol part of the practical.
Okay, thanks, Matt. And again, if anyone wants to follow along with sort of PyMol on another screen, we've provided you the PyMol session file in the data pack. So it's all there ready to go. If you want to sort of work through reproducing it offline, we've provided the commands in the cheat sheet and the structural model files for you to use. So here's one I prepared earlier. So just to walk you through a little bit what everything in here is before we sort of look at what's going on. The pink line shows the chain of the antigen. The orientation, the way that you can see it. The cyan line on the right hand side corresponds to the experimentally determined structure that we want to align our model to, and trying to align in such a way that you can see the differences. The green line corresponds to the model of our clone of our antibody that was generated using a body builder. And on top of that, the yellow line here shows the modeled modeled CDRH3 of our antibody, and the orange line here shows the modeled CDRL3 of our antibody. And so you'll recall that we found a sequence match for the heavy chain of our antibody, but we had to borrow a compatible light chain for our antibody because, thinking back to the original database, we only have a heavy chain. We don't have a light chain to go alongside it. But to get an idea of what's going on here, we can see that overall the structure and the model of our antibody align very nicely. The CDRH3s are where they should be. The CDRL3 is mostly where it should be, and I'll come back to that in just a moment. But the framework region surrounding it is very similar. And if we think back to what we were talking about at the beginning, the framework regions are sort of necessarily very conserved across different antibodies. They don't play a role in affinity or specificity. But just to give an idea of what the similarities and differences between the structure and the model might be, and how that might lead to a structural hypothesis of whether or not this is actually going to be a good binder or not. If we look first at the modeled CDRH3, so the binding portion of the heavy chain for our antibody, we can see that the yellow line and the cyan lines are very close together. So we've got the general position and conformation of the CDRH3 quite accurate. And what in particular you can see here, the sticks representation is highlighting residues that form part of the antibody's paratope. That is the portion of the antibody surface that is in contact with, is binding to the antigen. And one of the things that's interesting that immediately jumps out is the cysteine motif, or the cysteine bridge that Matt mentioned previously. Not only is this conserved between the crystal structure and the model that we have, but actually one of those cysteines forms part of the paratope. So by doing this modeling, we've actually been able to not just get things in roughly the right place, but actually get part of the paratope in the right place. And we can immediately see this conserved motif suggests that at least this part of the binding mechanism is going to be conserved across these two antibodies. So this is already starting to suggest to us that actually we have reasonable grounds for thinking this antibody could potentially bind in a way that we would like it to. However, where this starts to get a little bit different is when we look at the model of the CDRL3, so the CDR from the light chain. So again, the cyan is the crystal structure of this known binder, and the sticks representation here is glycine that forms part of the paratope. So in the solved structure, this is a part of the antibody that is going to be binding to the antigen. The orange here, however, shows the model that we've generated for the L chain that we borrowed for our antibody. And we can see that the model here is quite different to the solved structure of the known binder. In particular, unlike the known binder, there are no residues in this model that are close enough to the antigen to be involved in that binding. So we've generated a model where the CDRL3 doesn't actually contribute to the paratope at all. So that should get us to start thinking that well, it may not be the case. That this doesn't mean that that L3 is not going to form part of the paratope. If we had a slightly different model, that it might be more clear. But what this is certainly telling us is that it is likely that the antibody that we here is going to bind using the same interactions, the same binding mechanism as this solved structure that we're comparing it to. So, and if we sort of look at the sequence viewer up at the top of PyMol, we can see these orange residues here correspond to the modeled CDRL3. There are several insertions here, so there's a large gap here between the CDRL3 from the model and the CDRL3 from the structure that we've aligned it to. And you see that in this gap here, the CDRL3 isn't able to span over to the antigen in quite the same way as the structure that we're comparing it to. So overall, what we can really take from this is just from having generated this model, we can immediately look at it and see we're now starting to form a structural hypothesis for how this antibody might actually bind to SARS-CoV-2 spike protein. But also it's already revealing to us that, and again, this is where sort of identifying, these distinct novel sequences might be interesting. It may also interact in slightly different ways. It may have a slightly different paratope to the observed binder, and so we may need to think a little bit more about precisely how it binds. It may bind with slightly different affinity, slightly different specificity to this observed binder. But this sort of hopefully will start to give you a feel for how we can go from this sort of data processing and modeling pipeline through to actually forming this sort of structural hypothesis. So I'm going to stop there. You have the session available to you. Please do feel free to ask any questions about this, either live or afterwards. But we're now due for a 10-minute break. So I'll hand you back over to Chris, and we'll be back around 10 minutes past, where we'll get into sort of the more hands-on part of the workshop.
Okay, thanks very much. That's absolutely fabulous. It's really fascinating. Great to get this demo of a real hot topic as well. Bailey, I noticed that you've joined us. Do you want to say a couple of words?
Yeah, hi everyone. I'll turn that on now. First of all, I'd now just like to thank both Charlotte, Matt and Fergus for the excellent workshop that we've had so far. Just to introduce myself, I'm Bailey and I work with Liverpool ChiroChem. We're a chemical technology innovator on a mission to accelerate small molecule drug discovery through expanding access to 3D chemical space. So Liverpool ChiroChem is proud to sponsor this series of workshops, and as we understand how essential these open source tools are for global research communities. At LCC, we specialize in using stereo-defined N-heterocycles for using DEL, FBLD, bifunctional linkers, and our 3D discovery virtual library. If you would like to find out anything else about LCC and how we could support your projects, please reach out to us through our website, liverpoolchirochem.com, or find us on LinkedIn. I hope you all enjoy the rest of the workshop, and thank you very much.
Thanks very much, Bailey. So I'll put us on mute at the moment, and we'll be back in 10 past, as you said. See you in a little while.
Okay, think we'll start the next session now. If that works. Just so we don't drag into people's evenings. It's very warm outside, so
Yeah.
people want to have some fun outside. We understand that. Okay, so just really to consolidate and wrap up from the sort of things that Fergus was discussing, the idea that there might be structural similarities and sequence similarities between things that are functionally similar, but that they don't always align. Sometimes you have structurally similar things that have different sequences, and sometimes you have similar things with different structures. And just really to highlight the fact that we are developing a lot of functional clustering work in this space to think about ways we can combine sequence and structure into the way we predict whether two antibodies can do the same function, i.e. bind the same antigen. So we have a paratyping tool, which via prediction of which residues are in the paratype, they were the residues that were in stick representation on that pi mole, can calculate sequence identity just across these regions. So only the regions that we think are going to be involved in the binding. So that's like a sequence alternative to clonotyping, which we use to implicate the convergent clones. We also have a tool called obligity, which can couple this with a model of the antibody structure and then builds up an idea about the spatial disposition, the relative spatial orientation of these predicted paratype residues. And via hash table comparison can compare antibodies to one another to see if they would have the right properties. SAR+, which is more of a rapid structural annotation tool, can be used for similar purposes. And if you want to go higher level towards kind of docking and rescoring with deep learning methods, we also have work in that area as well, to see whether we can get a putative pose with good enough energy via the rescoring. So we have all of these tools, all open source on our GitHub that you could play around with if you're interested in this area. But for the rest of this session, we're going to be focusing on developability. So what we'll first do is just introduce the broad field of developability issues, then talk you through two of our tools that attempt to predict whether an antibody is more developable or less developable than another one. And then we have a series of tasks that you can go and work through for 15 minutes, or you can feel free to play with the web applications however you want. We can then reconvene afterwards. We'll show you a few of those tasks in action with a live demonstration. And then we'll wrap up and reflect on the sort of lessons that we've seen through the workshop and how OPIG tools can help improve antibody analysis. So with these developability issues, as Charlotte said, really, these are everything that is important to drug discovery that isn't the bind or not bind problem. So the first step is, does my antibody engage the antigen? But once you know it can engage the antigen, you'll have evidence that you think this would engage the antigen. You then also care about a whole range of other properties, such as, will my molecule aggregate if it's kept in a vial? Will it create a solution that's too viscous for injection and for handling, ease of handling? What's the immunogenicity like of the antibody? So if you had developed this in mouse hybridoma, for example, then to some extent most humans would be expected to recognize this as a foreign molecule, and so you have to think about perhaps how you can alter its sequence to make it seem more human-like. Maybe the antibody does bind to the target of interest, but it also cross-reacts with human proteins that we don't want to happen. And it's possible also that, as can often be seen by poor expression titers, there's some inherent instability to the antibody. That could be chemical instability, that could be structural instability. It's kind of making the antibody reluctant to fold properly, and you end up with a poor drug as a result of that. So our tools that I'm going to present now sit in this space, not predicting all of these developability issues, but trying to predict some of them. And the first one I'll describe as the therapeutic antibody profiler, or TAP. So what this is really trying to detect is whether an antibody model surface has atypical biophysical properties, so atypical distributions of, say, hydrophobicity or charge properties, unusually high patches of charge. If we think they are unusually kind of condensed, we would say that this is probably not good from a therapeutic development perspective. So the way it works pipeline sense is it uses a bodybuilder to generate a homology model of your antibody, and then it scans the surface, calculating certain surface properties that are linked with aggregation, viscosity, instability, polyspecificity. In a way, it's our version of doing the Lipinski rule of 5, but whilst small molecule structures may be easier to get a handle on what they might look like in 3 dimensions, antibodies are more complicated, so you have to go through this explicit modeling pipeline and then calculate these properties on the model. So We have packets of hydrophobicity is one metric we use a lot, positive and negative charge, whether the two chains have a significant asymmetry in the way that they are. One is very positively charged, one is very negatively charged, which has been associated with drift of the antibodies together and associated viscosity, and also atypical CDR lengths. So you can think of this in two ways. If your CDR lengths are all really short, really low, you have a very featureless binding site, or you probably have a featureless binding site, which might mean that your antibody is quite promiscuous. It can bind to lots of different things. On the other hand, if you have an antibody with a really long CDRH2, this is protruding from the surface. It could be a site of aggregation. It could be a site of structural instability. It might bind to lots of different things by virtue of having lots of low energy conformations. So this really starts to capture things that are correlated to developability and seeing it kind of by proxy. And what we use to define, is this property too bad or fine, is a reference set of therapeutic antibodies that have made it to at least phase 2 of clinical trials. So by this point, it's assumed that these antibodies are probably broadly developable, because if a company has invested past phase 2, it's likely that these more kind of engineering issues might have either been worked out or never cropped up in the first place. And these are a reference set that are shown in blue as a histogram for a given property. And then if your inputted sequence falls within the range that most of these therapeutic antibodies do, you get a green flag. If it's starting to get towards the tail ends of the distribution in a way that we predict would be deleterious, you get an amber flag. And if it's unlike any therapeutic we've seen before, you get a red flag. And the web application itself, quite similar to the ones you've seen us use before. So you input the whole FV sequence of the antibody you're interested in profiling. So that's the heavy chain / the light chain in amino acid. So it's / limited. And what you can expect out is a table with the flags and the values associated with each property. And you can see those in a more intuitive sense with the histograms. 5 histograms are displayed for the 5 different properties. And you also have a molecular viewer. So you can see, for example, if you color by hydrophobicity of different residues or by positive and negative charge, how those features are distributed across the surface and why the antibody might have got the score it did, which is very useful from an interpretability perspective, because it allows you to then think, okay, maybe I could make these molecular changes and improve my developability as well. And Fergus will talk you through the humanist side of things.
Okay, thanks, Matt. So in addition to thinking about sort of the developability of our antibodies, so Charlotte, if we think way back to Charlotte's introduction, even then we were thinking about this idea of it doesn't matter how strong and specific of a binder our antibody is if, for example, it's going to aggregate in storage. So we need to be thinking about just purely in terms of given an antibody, can we deliver it safely as a therapeutic? But sort of as Matt and Charlotte both alluded to, sorry, Matt, can we go back, please? Thank you. As alluded to previously, it's not enough to just think about, for example, as a small molecule, it's not enough to just think about is this thing, is it easy to transport? Is it easy to distribute? Is it going to aggregate? Is it going to have a good shelf life? Just like in small molecules, where we have to think about things like toxicity. And off-target effects. In antibodies, we have to think about, how is our immune system going to respond to this, potentially very novel antibody sequence that we're injecting into our system. If we put something that doesn't look like something the immune system recognizes as one of its own antibodies into us, is it going to raise an immune response against that antibody and try to get rid of it, or potentially cause horrible effects for us? And this is known as immunogenicity. It's raising an immune response that we don't want. And so if we're thinking about this in terms of developing a potentially novel antibody, we might start from something that is human. So in this case, we started from human antibodies for SARS-CoV-2 from the OAS database. But in order to optimize affinity or developability, it's often necessary to make mutations to the sequences, so to change some of the amino acids in the sequences. For example, if you think back to the Pymol session we looked at earlier, we noticed that the CDRL3 of our lead candidate wasn't actually in contact with the receptor binding domain of the SARS-CoV-2 spike protein. And so the CDRL3 will likely need to be mutated in order to be elongated in order to achieve stronger binding, in order to actually bind in the way that we observed with the experimentally solved structure. And this is obviously this is going to be necessary in order to make a good antibody, but in the process of doing this, we may result in a sequence that no longer looks entirely human. We borrowed a light chain that is human, but by making changes to it, it may come to more closely resemble, for example, a mouse sequence, which our immune system is going to be less happy with. And so before we even think about trying to solve this problem, we just need to first be able to predict whether or not a sequence actually looks human, because this alone will allow us to anticipate potential issues with immunogenicity. And then further, if we conclude that our sequence does in fact no longer look human, we need a way to humanize it again. Next slide, please, Matt. So humanizing a sequence is something that is actually done very commonly experimentally. But one of the problems is it's often not entirely clear what mutations would be best to make. For example, we can look at the CDR-L3 and say, okay, this L3 needs to be a bit longer, and maybe we might expect to see a certain residue in there. But really, it's often not entirely clear where to start making those mutations. So experimental mutation campaigns can often be sort of very exhaustive. Let's just enumerate all of the possible mutations that we could make, or a very large number of them, and then just assay everything and see, of the mutations that we've made, do any of them bind better than what we started with? Obviously, this will converge upon the solution if there is a solution, but it's very costly, it's very time consuming. And it doesn't really solve this problem of, can I look at this and say, does it still look human? You might end up mutating even further away from human, chasing those other properties. So in order to try and address this problem rationally and computationally, we've developed a tool called HUMAB, which is a human antibody humanization tool. And this tool really does two things. So at the core of this tool, there are a set of random forest classifier models that have been trained on essentially all of the human and mouse light and heavy chain sequences that we were able to extract from the OAS database. That was a lot of sequences. I mean, there's around 60 million human sequences and around 15-20 million mouse sequences. There are also some rat and rhesus monkey sequences in there, but the overwhelming majority of non-human ones were mouse ones, just because that's what's represented in the data. And so the core of this is a set of random forest models that have been trained on these sequences in order to recognize whether a sequence is human or not human. And actually, it turns out that the most rational way to do this is to use a different model for a different V gene type. So your light chain and your heavy chain will come from will map back onto one of a very small number of initial V genes. And so by making that distinction, you're actually able to get much more accurate predictions about, does this actually look like a human sequence from this particular V gene origin, rather than just taking the naive approach of throwing in all of the data into a single model. And now the end result of this is a classifier that will assign a score to how likely it thinks that this sequence is actually human. And you can threshold the score in such a way that you obtain an accurate classifier that says, yes, this is human, no, it's not human. And it turns out that for some V gene types, you require much greater confidence than others in order to actually say this is human. So again, this brings us back to this idea of, we need to be able to identify V genes to do this rationally. So that lets us predict whether a sequence is human or not. But what we found is that we are actually able to extend this to not only predicting whether a sequence is human, but actually to developing an iterative computational procedure for taking a initially non-human sequence and mutating it to something that looks human, that passes the classifier and looks human. So the way we do this, if we look at the bottom schematic, is if we start from some initial sequence and we assign an initial humanness score, this will tell us, it's not human, but we think this is good. This is the V gene type. And what we can then do is go through an iterative sequence of mutations. So we pick a residue, we mutate it, and we rescore it. And if it looks more human, we keep that mutation. If it doesn't, we discard that mutation. And so we can iteratively, very quickly, certainly much more quickly than doing an experimental mutation campaign, we can iteratively go through this process of making mutations, assessing whether the sequence looks sufficiently human, and continuing to do so until we arrive at a sequence that we consider to be sufficiently human that we would be happy to proceed with it. And again, thinking about doing this rationally, the way we approach this is we make these mutations only to the framework region. So we leave the CDR regions in the sequence completely unchanged. And by doing this, what we're able to do is take the sequence and turn it into something that looks a little bit more human by making the framework region look human, but keep that incredibly diverse CDR loop that is responsible for binding and specificity, keep that unchanged, and so as best as possible preserve its binding properties. And so, yeah, next slide, please. Thank you, Matt. So the link to, as with everything, the link to HuMab is in your cheat sheet. You're going to have an opportunity to play with this in just a minute, but just give you an idea of what HuMab is actually going to generate as output, what it's going to look like. If we feed in a sequence, in this case, we fed in a sequence that is human, what you'll generate as output when you perform the humanist classification is it will, for each of the possible different V gene types, HuMab will score the sequence according to, it will feed the sequence into each of its classifiers, and so it'll simultaneously check what do I think the V gene of this sequence is, but also, given that, do I think this is human? And what we can see here is that actually HuMab was able to identify that it is a HV4 sequence. And given that it's a HV4 sequence, actually this looks like a human sequence. And so typically what you'll find is that it'll be very clear. It's very clear that it is from a particular V gene sequence. So we don't really have this ambiguity of I'm not entirely sure what the V gene is. It's very, it's usually very clear cut. And next slide, Matt. And so then thinking further now about, okay, what if I had a sequence that HuMab thought wasn't human? What does this humanization protocol look like? If you'd said, and this is on the same page as the classification tool, if we instead feed our sequence, and we can do this for a heavy chain or a light chain, or both a heavy and light chain at the same time. If we can feed in our sequence to the humanization protocol, this will take that sequence and compute its initial humanist score. And if it thinks it's not human, it will step through that iterative protocol that I described by making these sort of sequential mutations and continuing to do so until it arrives at something that it classifies as human. This protocol can be very quick if your sequence is very close to human. Maybe it only takes a single mutation to make it human. That might run in, say, 30 seconds. However, if your sequence is quite far from human, this protocol unfortunately can take some time. It can easily take maybe 15 minutes for something that is, very much looks like a mouse sequence. So we're not going to be playing too much with the humanization protocol today. However, we will sort of show you what this looks like. But just get a feel for what actually happens here. Again, thinking about this rationally, when we humanize a sequence in HuMab, we're able to have a look at what individual mutations were made in order to make this look like a human sequence. So we can see in the output down here, we have our initial sequence with the IMGT, thinking back to sequence numbering, we have the IMGT residue numberings at the top of the sequence. The dark blue in this display corresponds to a framework region. If you recall, we're only mutating the framework region. The light blue corresponds to the CDR loops, which is where your binding properties come from. And you'll see that no mutations have been made to the binding loops. But you can see in this case, we have annotated quite a few mutations have been made just to this portion of the framework region. And it is quite common to actually make quite a large number of mutations. We can see here, ultimately 16 mutations were made. So 16 individual amino acids were changed to something else in order to get the sequence to look human. Obviously, you could continue this procedure further to get something that looks even more human, but realistically, you want to be making as few mutations as possible. And so this will sort of iteratively go through until it gets something that looks human enough, and then stop, and then report back to you on exactly what has been mutated. And with that, I'm going to hand you back to Matt, who is going to describe the practical that you're going to be doing for the rest of this workshop.
Thanks, Fergus. Yeah, and another point I think you just briefly touched on was that that HuMab, every time you make a mutation, you don't fully know what influence that's going to have on the angle of the binding site. So HUMAB does try and also make the fewest number of mutations it can. So each of these mutations has a directionality to it. So it's going to make the mutation that it thinks increases the humanness by the most first, and then go from there. It might also be more efficient than iterative kind of experimental methods, where you might end up making a lot of unnecessary mutations to your sequence, which at every point you do that mutation, it could have a knock-on effect. So yeah, so those are our two tools that do the a lot of developability assessment. So now we're going to allow you to have a more of a workshop experience and go away and play with these web applications, the URLs to which are in your cheat sheet, as well as you have the sequences for each of these different things that are underlined as FASTA files in the resources as well. So some ideas of what you might want to do is hunting in that convergent clone that we found way back in the first half of this, and see whether TAP thinks it's developable across its 5 different characteristics. And also, does HUMAB think it's human? I mean, we took it from a human consensus sequence, so you would hope so, but does HUMAB think it's human? We've also provided a sequence of another antibody from COABDAB that was found by phage display rather than fishing from humans. And you can really see the influence of perhaps what sort of in vitro technologies can do to developability, because there's no selection for developability when you're making these libraries. And TAP predicts that this antibody will have a very atypical biophysical property. So you could stick that sequence in of MERS93 and have a look at what TAP finds problematic about it. Also a nice little potential case study if you want to go to COABDAB, and find things that have been experimentally humanized from immunized mouse, and you can compare that with HUMAB's humanness scoring. So an example that we've already picked out that has a really nice example for this is HB27. So you could see, does HUMAB consider both chains to be human or not? What's the threshold of that? And then it might take longer than you have before the end of the little workshop, but you'll see if you feed it into the mutation suggestion tool, which mutations or how many mutations HUMAB thinks that whichever chain is non-human requires. And you could also then think about this cycle of, you've made these mutations, but now has that affected my biophysical characteristics? And put the mutant and wild type sequence back into TAP and just make sure that isn't the case. So these are just some ideas for you to play around with, but it's completely up to you really to do what you want to do. We're going to be monitoring the chat. So if you have any questions about how to use any of the web apps or what you're seeing from the web apps, feel free to stick the questions in there. Other than that, we'll reconvene perhaps at between 1:45 and 10:45 for a little wrap up, and walk through some of the things that you might have seen. And then we'll consolidate and end, or we're happy to stay on and answer questions after that too for a little while. So with that, I'll stop sharing the screen and allow you to play with our tools. I should mention as well that the PDF of the presentation is also in the resources. So if you want to look again at those potential case studies or things like that, then the resource is there for you to see what suggestions we have.
So I have another question about AlphaFold for you. I've seen some people do measure or model protein-protein interactions using AlphaFold by providing a very long flexible linker between proteins. Have you tried that with an antigen and an antibody yet?
I actually shared the same, I saw this on Twitter, I shared the same thing with our group just a couple of days ago. But not yet, but I think this is something that we're seeing if you can game this a little bit by using this long glycine linker, not just for antibody antigen, but also potentially for VH and VL, which of course aren't covalently linked, they're just non-electrostatically linked. So that could again be a way of doing complete rather than single domain modeling, actually being able to model the full FE domain at one go. But yeah, have noticed that hack and keen to see if it actually works out.
Yeah.
One of the benefits perhaps from making things open source is that people take things and think creatively about how you could use them.
Yeah, absolutely. I mean, I think it's I mean, I come from small molecule drug discovery, so I find all these things a bit like black magic at times. So I do find them fascinating, though.
Yeah, it's really great that we've got access to the source of this, and we can go digging to how it's getting this performance. And one of the things that we don't know really whether it will necessarily translate well to antibody modeling is because it borrows a lot from, it learns a lot with an attention on the multiple sequence alignment, which is not a classical approach that people have used to model antibodies because of the sequence variability as well as gaps coming in odd regions of the sequence. So it might be that it just needs a slight change of perspective for an antibody specific tool, but that a similar approach could work for antibodies. Might need a bespoke tool basically. Or it could be that MSAs work well across particular regions, and that we should use this sort of modularity. So that's something that we're certainly interested in and looking into, because we're always trying to get better structural representations, because it means that all the tools that we've built on top of them will will be able to find use and be more useful for people in industry.
And things like your humanizing tool and things like this, do you constantly update it with as you get new sequences available?
So actually, Fergus probably is the better person to answer this, because he moderates and maintains our web applications. But certainly for the antibody database, it's an auto-updating. The structural antibody database is auto-updating, as are some of our other databases. But so the templates that are abstracted from that to build the models, they also reflect the latest structural knowledge. But I don't think the Humanus random forests are rebuilt every time we add a data set to OAS, but I think it can be triggered. It can be, maybe it's something that if we think there's been a lot of data added, we can rerun that. But I think our accuracy was really quite high. We were getting ROC AUCs towards 1, so I don't know how much improvements of performance this will actually give us. But Fergus, if you want to speak to that.
Yeah, I mean, I think you've sort of, especially with the HUMAB stuff, I think you've sort of pretty much hit the nail on the head there. If an enormous amount of data came out that we thought might be beneficial to bake into HUMAB, then we could certainly rebuild the classifiers that it used. Or if, for example, we suddenly discovered through maybe in the next pandemic, we weren't actually very good at recognizing a certain set of antibodies as actually being human, then that might trigger the need to reevaluate. Do we need to retrain this with some new data? But with the way things are currently set up, the accuracies are sort of sufficiently high that we sort of keep the models as is, and it would very much be a deliberate decision to update them, because again, when we're sort of thinking about things in sequence space rather than structure space, which is what HUMAB was trained on, we're suddenly thinking about the scale of the data involved. If you have a database of a few thousand antibody structures, and you update it with 100 antibody structures at a time, then sort of auto-updating that and auto-updating software that depends on it is really straightforward. However, if you have a database like OAS, where you have 80 million sequences, and you add 5 million more sequences, that suddenly actually becomes quite a large undertaking to sort of, and the sort of very infrequent, irregular, but very large and very heterogeneous updates in terms of size.
Not all
so
sequences are made equal.
Yeah,
so one thing that HUMAB does much better is distinguishing human from mouse, because that's what the bulk of our data is. If we suddenly got a whole lot of rabbit data, that could tell us a lot of information, even though it's not many sequences. That could tell us an entire new way not to be human. And so these sorts of things, we're always on the lookout to see if we get a bulk of data from different sequences.
Exactly.
Species, sorry.
Yeah, exactly. I think that the interesting thing to do with HUMAB would be, rather than updating the human mouse classifiers, unless we start to think, okay, maybe they're not as good as they could be, it would be introducing new classifiers that say, do I think this is mouse? Do I think this is rabbit? Do I think this is rhesus monkey? What do I think this is? Because you can imagine where this becomes relevant is if you're sort of doing this mutation process. If you're sort of trying to think about doing this mutation in an intelligent way rather than just sort of throwing the kitchen sink at it, you might zone in on sort of different starting points if you're starting with a rabbit sequence as opposed to if you're starting with a mouse sequence.
I guess another sort of, in some way, quite scary thought is the fact that we may get to a situation with COVID where sort of domestic animals and companion animals become a reservoir for the virus. And you might say, well, can we design antibodies against those as well to inoculate the pets?
Absolutely.
Yeah,
pet medicine is it? Sorry, folks.
I was going to say, absolutely. That would be sort of and again, this kind of feeds into I mean, we're familiar with this in computational chemistry. There's enormous bias in terms of we have more binding assays for compounds that the medicinal chemists want to make than we do for the ones that they don't want to make. Yeah. In much the same way, if it suddenly becomes incredibly relevant to have, if it turns out dogs can catch and spread something like COVID-19, you might suddenly have a sequencing campaign of dogs.
Yeah.
And that's usually what triggers this, right?
Yeah. And that is a sort of, that is a form of data that just up until that point will not have existed, for COVID-19 response or otherwise. And so in and of itself is a really interesting thing to add in, but certainly would be, vitally important for actually, as you say, it's not just enough to inoculate humans. And, we need a way of designing antibodies, for other species as well.
Yeah.
Great. So I think I'll just go through the last little bits of live demo that we had planned for you. And then we can wrap up and discuss what we did. So I'm here on the TAP homepage, hopefully you can see. And what I'm going to do is I'm going to profile the Convergent clone. So the Convergent clone that we saw across 3 COVID patients, but not in other diseases, and run it through TAP to see how developable it is from those biophysical perspectives. So this will first of all create an A-bodybuilder model. So many of the first steps are just A-bodybuilder going through its thing. And thereafter, it will calculate the patches of surface hydrophobicity, so regions where the molecules might aggregate through exclusion of water from the interface of particular exposed hydrophobic patches, charge patches, either positive or negative, CDR length distributions, and charge asymmetry. So what you can see here is that all 5 properties it checks out. So it's not always the case that human antibodies are nicely developable like this. In fact, the TAP paper itself is somewhat predicated on showing you that the distributions of these properties are different for therapeutics than general human antibodies. But in this case, it looks like we've lucked out quite nicely. So we've got 5 green flags here. And then if we look down at the individual properties, you can see the CDR length is really close to the median of this distribution, as is the hydrophobicity. There are no obvious patches of positive or negative charge shown by these green lines here. And the charge asymmetry, actually, this is calculated by taking the net charge on the heavy chain, the net charge on the light chain, and multiplying them together. It looks like they have a net charge on both chains that is of the same polarity. So they're not going to be coming together. Each unit is not naturally likely to be drifting together just by virtue of its net charge. And we have our model down here of what this antibody looks like in 3 dimensions. And just to verify here, where red is more hydrophobic and blue is more hydrophilic, we can see that there's not many patches of red, really, if any, on this surface. Nothing that the antibody might be expected to come together and exclude. And the same thing for the charges. Blue are negatively charged residues here, red are positively charged, and you can see that they exist really quite nicely, evenly spaced out across the molecule. So nowhere where they're going to be sort of constraining conformational diversity and sort of decreasing the stability of the fold, and equally nowhere where there's going to be a blob of negative charge that's going to bind to anything positively charged in the body. So that's the tap representation. So now I'll just flick over to HuMab to see if the same thing can be said for its humanist characteristics. So I'll paste in its heavy chain sequence and its light chain sequence. Helpfully, our web applications sometimes take these as a joint input and sometimes don't. But first of all, we'll just score the humanist of this sequence. So again, this is going to pass the sequence through the random forest classifier. It's going to work out, it's going to automatically detect which is the heavy chain and which is the light chain. And then based on its different thresholds and classifiers for the different V genes, it's going to say, is this a human sequence or not, for each of these chains. So it's already finished. So it's decided with very high confidence that this is a human HV1 gene derived sequence. Big gap there from the threshold. So it's a human heavy chain for sure. And the light chain we borrowed from our in-house library. You might assume that we wouldn't just want to introduce non-human stuff from it. And this also came from a human antibody, just a different one. And that also has you'll notice in the light chain there are slightly tighter thresholds than the heavy chain, but again, blew it out of the water, 100%. Confident really that this is a human sequence and that it comes from the kappa locus, not the lambda locus. You can download these results and get a log file here for the job. And like I say, you could also, it's a bit pointless doing this because both chains are 100% human, but if you then wanted to go around how best ought I humanize my sequence, you could use these query boxes here, which Fergus will talk you through the results of having put the HB27 antibody, certainly the light chain of that antibody, which was actually scored as non-human by HuMab despite experimental humanization. Fergus will now walk you through the results from what you would see if you'd done that humanist mutation profile on that antibody. Oh, sorry, I stopped sharing. Should have done that. Let's go back. Sorry, Fergus, take it away.
Thanks, Matt. Yeah, so again, thinking back to our previous description of HuMab, when you run a sequence through HuMab, what you'll actually be presented with is not just a summary of the number of mutations made and where they were made, but actually if we look at the job log at the very top of the screen, we can actually see the order in which these mutations were made and how these mutations contributed to the increasing humanist score. So you can see that we started with a humanist score of 0.74, and for this particular V gene type, we're actually aiming for a humanist score of about 0.84 in order to that is what would see it regarded as human. So we can see we're actually reasonably close. It's not too far off of looking human. And so in this case, you can see that in this case, in fact, only 3 mutations needed to be made. And you can see that each of these mutations actually improves on that humanist, quite significantly. And so again, thinking back now to what the sequence actually looked like, it looked like it was close to being a binder anyway. So we might intuitively expect that in sequence space, and again, we have this hypothesis that we could start to see areas in the sequence that mapped onto functional differences between how the antibody behaves. From that, we might expect that it will only take a few mutations, but this gives us a nice confirmation that actually we were quite close to having something human, and it was easy to make those mutations. But what we can see here is we have a summary of where these mutations were made. So in this case, we needed 2 mutations in framework region L2 and 1 mutation in framework region L3. And again, thinking back to, we were only making these mutations in the framework region, so we don't expect and we'll never see a mutation in the CDR region. So while these mutations might have some impact on the overall structure of the antibody, they really should have a minimal impact on the actual binding profile of the antibody, because those CDRs will be completely conserved. And so hopefully what we can see from this is that we're able to take our sequence that we think is going to be a good binder and actually mutate it into something that our immune system should recognize as human, and therefore not be less likely to have immunogenicity effects, while preserving the affinity and specificity of that antibody. We can think of this, if we want to think of this in chemistry terms, we can sort of think of this in terms of we're now going through the optimization process of a small molecule drug. We want to, once we've achieved affinity, we want to optimize things like specificity, toxicity, metabolism, etc., all of your ADMET properties, while preserving that. We're not necessarily trying to achieve more affinity. We just want to preserve what's already there. And so now, just to briefly summarize what we've gone through in this workshop. Again, you have the cheat sheet there if you'd like to work through this again offline. Starting with our OAS database, we found a data set of antibody sequences from COVID-19 patients, and the patient metadata preserved in the database enabled us to compare the sequences raised across the 3 individuals. And so what we did in this workshop is we compared the heavy chain clonal lineages across all 3 individuals, finding 2 public clones, that is sequences that are raised in all 3 individuals. So they're sort of evolutionarily converged upon. One of these sequences turned out to be present in many other unrelated disease data sets, suggesting that it's part of a background immune response rather than something that is going to be a specific binder for SARS-CoV-2 spike protein. So we focused on the sequence that was only found in the COVID-19 patients. From there, we took the CDRH3 loop sequence, recalling this is the sort of very variable region that's heavily involved in binding of our convergent clone, and compared and queried this in CovAbdab, which led us to a very sequence similar antibody, which actually had a solved structuring complex with antigen in the PDB. So this starts to allow us to think, actually, this may actually form the basis of a good binder. We then borrowed a compatible light chain. Again, we only have a heavy chain here. We borrowed a compatible light chain from one of these databases that should pair with this heavy chain, and modeled this VH-VL pair using the A-Bodybuilder pipeline. And thinking back to our PyMol session, when we aligned this to our solved structure of a SARS-CoV-2 binder, we found that the topology of the VH was very similar to the solved structure. In particular, there was a cysteine bridge motif that was both conserved and part of the paratope, so the antigen binding region. And so it appears to be compatible with the same SARS-CoV-2 binding mode. However, we also observed that the VL chain that was borrowed deviated a little bit, and so would likely require some additional mutation in order to make it a more optimal binder. And so, but given this evidence and structural hypothesis for the binding behavior, we then examined the potential developability issues of this antibody as a drug using TAP, which allows us to do this biophysical characterization of, does this look like approved therapeutics, or are there some properties that are sort of out with the space of what we would normally observe that might set alarm bells ringing? And then further thinking about, once again, we need to make mutations to this sequence in order to make it an optimal binder. We were then able to use HUMAB in order to assess the humanness of our sequence. And in this case, HUMAB raised no issues with this particular clone. That was quite fortuitous. And so with all of this sequence-based and structural-based information taken together, along with these considerations of developability, what we've arrived at in a relatively short space of time is what appears to be a very strong candidate for further preclinical development. So next slide, Matt. And so the take homes from all of this is that this isn't really purely a structure-based or a sequence-based problem. And in fact, using both sequence and structure-based information together as part of your early stage antibody therapeutic development pipeline can really help us to better understand antibody function, allow us to sort of more quickly arrive at good potential antibody therapeutic candidates. And so over the years, we and others in OPIG have developed numerous tools and databases to facilitate this, in particular for generating structural models from sequences and how to learn from the structure representation. Again, thinking back, solving structures experimentally is never going to keep up with the glut of data coming in through next generation sequencing. And so in order to build these structural hypotheses, we really want to be focusing on generating the highest quality models that we can in a tractable period of time. And in OPIG, in order to facilitate all of this, we've also created several databases that are both useful as a source of raw data, just having all of the NGS data in one location, but also as data points for cross-referencing of data. So again, thinking back to what Matt showed you earlier, we can immediately jump from a sequence in OAS to actually looking in CovAbDab to see, do we actually have some known COVID-19 antibodies in there? Jump from there to SABDAB to, do we actually have an experimentally determined structure of this antibody? And this allows you to sort of very quickly go from an initial starting point to actually building up this body of structural and sequence-based data that you might use for further development. And so really the crux of all of this is that these sorts of tools are especially useful in early stage drug discovery, so lead prioritization. I believe someone very early on asked the question about using, for example, molecular dynamics in this pipeline. What I really want to stress is that this sort of workflow is very much the sort of workflow that you would use in the early stages of drug discovery. So you're trying to do very high throughput screening and analysis of sort of very large amounts of data in order to bring this down to a sort of a tractable number of potential therapeutic candidates, and then examine those candidates to see whether it would be worth taking them onto preclinical and clinical development. And so much like this problem of attrition in small molecule drug discovery, by using these sorts of tools in antibody therapeutic discovery, you can really reduce the time and resources that are spent in this early stage drug discovery process, and maximize the number of candidates that you would be taking forward to preclinical development. And finally, as I mentioned right at the very start, you have in the cheat sheet links, download links to all of the tools that we have available, and also links to all of the web servers, also references for the papers if you're interested. But just to highlight the availability of the software and source code, many of our tools are available on our GitHub page at OXPIG. However, a large number of the tools, in particular A-Body Builder, HUMAB, TAP, the other components of the SABPRED platform, as well as the SABDAB database, because of their complex interdependency with each other, as Matt stressed, A-Body Builder is very much a modeling pipeline. We don't distribute these tools individually because you really need sort of the entire setup in order to use any of them effectively. So instead, to deal with this, what we instead distribute is a virtual machine, which contains the entire SABDAB database and SABPRED platform pre-configured and ready to use. And this is available. It's available completely free of charge under an academic license from the Oxford University Innovation Software Store. Links on the slides in the cheat sheet. If you're an industrial user, it does come with a license fee, but it is exactly the same version that we would distribute under the academic license. And with this virtual machine, you can take the entire platform, run it on any system, and it's ready to go. It does also come with the entire web server setup, so you can run all of these web apps locally as well if that's your preferred way of interacting with them. So we'd just like to wrap it up there. I'd like to thank Chris for inviting us to come and give this workshop today. I'd like to thank all of you for listening, especially if you're somewhere where the temperatures are a little bit sweltering today. 2-hour workshops can be quite intense. So thank you all for coming, and we'd be happy to answer any questions you might have.
I suspect many people have blocked out just these 2 hours, so we're right at the limit now, I think. But I think you've actually given us real tour de force of the capabilities of the. I guess it's the fact that you've got all of the software, the databases, and everything interlinked, and all the data well curated as well, of course. That makes it such a really powerful platform. I've been really impressed with it. It looks really fantastic. I'll have to have a play later on. So I'm getting a few messages to me saying to pass on thanks to you guys for the presentation. It's obviously that people have really enjoyed it, and I'd like to, on behalf of everybody, thank you for giving us a fabulous 2-hour presentation.
Thank you for the opportunity and for allowing us to walk through our last 4 years of our lives. Just to reiterate that, yeah, we've run out of time today, but we're very happy to answer questions by email. If you just ping us an email to opig@stats and you want to inquire either about the availability or the licensing or just how any of our tools work, always happy to receive emails. So just feel free. Thank you again.
Well, once again, thank you. And I think you both deserve a well-deserved cool drink now of some kind. Okay, right. Thanks very much then. See you. Bye.
Thanks. Bye.
Bye.